Okay, well, I first want to start off by thanking the organizers. For those of you that don't know me, I did my graduate work with Brian Crane at Cornell University, where I studied bacterial chemoreceptors. And then I moved to Yusuf Hanoun's lab, where I studied the structure and function of sphingolipid metabolizing enzymes. So it's nice to see a lot of friendly faces in the audiences. I'm going to tell you about what my new lab has been studying. I've been a professor at Stony Brook for about 3 years, and we've branched out a little bit to phospholipids, sterols, and glycolipids, but we're also still interested in sphingolipids. So today I'll tell you about some work on lipin phosphatidic acid phosphatases. Okay, so before I start, I just want to highlight the people that did the work. Weijing Gu, who's a master's student, Valerie Cahill, who's a PhD candidate, and Xuzhuan Gao, who's a research scientist, and she won't give me her picture, but that's the best likeness I could find on the internet. I also want to thank our collaborators, Karen Rui, Wan Wang, postdoc in our lab, and then John Burke for his help. So for those of you who aren't familiar with lipins, they are phosphatidic acid phosphatases. But before we knew that they were enzymes, Karen Rui's lab actually found that they were important enzymes in fat storage and lipid metabolism. And she did this by studying a mouse called fatty liver dystrophy mouse. And they had an interesting phenotype where their adipocytes never differentiated. And this led to a loss of adipose tissue in their fat pad. And her lab was able to show that this resulted from a single point mutation in lipid glycine to arginine mutation in a domain that we'll talk about later called the NLIP domain. Her lab also did the opposite experiment where they engineered transgenic mice, overexpressing lipin in both skeletal muscle and adipose tissue. And in both of these tissues, the mice became severely obese, with differential effects on insulin sensitivity and resistance. It was a few years later that George Carman and Jill Suhan showed that lipin was actually that missing link, the missing phosphatidic acid phosphatase in the glycerol-3 phosphate pathway. So this is also known as the Kennedy pathway, and if you're not familiar with it, it happens in the endoplasmic reticulum. Glycerol-3 phosphate is sequentially acylated by two enzymes, GPAT and LPAT, to form phosphatidic acid. Lipin, which is the only peripheral membrane enzyme in this pathway that has to associate with the ER membranes, then dephosphorylates phosphatidic acid to make diacylglycerol, which can then be acylated by DGAT enzymes to make triacylglycerol. And so it was this finding that this was an enzyme in phosphatidic acid phosphatase that actually explained why we saw these effects on fat storage. But its function is not just limited to triglycerides. Phosphatidic acid and diacylglycerol are both precursor for all the different phospholipids, and they can function as cellular signaling molecules to activate mTOR and PKC. And so there's been a lot of studies at both the cellular and physiological levels establishing lipin as a key player in lipid metabolism. So for us, we've been really interested in understanding how do lipins function as enzymes. And what you're looking at here is a domain architecture of lipin. There's two conserved domains. One is called the NLIP and one is called the CLIP. And the protein is about 900 residues, and these two domains are located on opposite ends of the molecule. So we know that the CLIP is shown in pink. It belongs to a large family of phosphatases. This is the codes for the catalytic domain, and it has a conserved motif called the DX-DXT motif. And so we know this is where the dephosphorylation reaction would happen. The NLIP is quite interesting. This is the most conserved region in lipins. It's only found in lipin proteins. And its sequence and structure and function is really unknown before our work. But we knew it was important for enzyme activity because that mutation in the fatty liver dystrophy mouse was within the NLIP. And so the major question that we're trying to understand was how does this NLIP communicate with the CLIP to form a functional enzyme? And the approach that we tried to use was a structural biology approach. And the problem that we had was that this separating these two domains was about 500 amino acid linker that was hyperphosphorylated and controls the localization and the activity of lipins. But it's predicted to mostly be intrinsically disordered. So for a structural biologist, this is a nightmare. It makes it very difficult to structurally characterize this enzyme. So we hypothesized that there might actually be unregulated lipins that lack this regulatory linker, and they might directly fuse these two domains, where the N-lip and C-lip are directly linked with each other. And so what we did was we took the sequence of human lipin 1 and directly artificially fused the N and C-lip and did a blast search and looked for endogenous proteins that might have this architecture. And we found about 10 of them, mainly in different organisms. They were mainly in plants: tomato, spicy pepper, potatoes, tobacco plants. And we also found them in some ciliates, and the one that Valerie had success with was from tetrahymena thermophila. And so this enzyme is called Pah2 or Pah2 for PA hydrolase, and this is sort of the nomenclature that we use for the yeast enzymes. So finding these sort of unique domain architectures, the first thing we wanted to do is test if they're functional inside of cells. And we took advantage of an easy phenotype to screen in S. cerevisiae, where you delete the lipin homolog Pah1. And these cells grow fine on glucose, but when you give the non-fermentable carbon source glycerol, they do not grow very well. And you can reconstitute that growth if you add back the wild type S. cerevisiae Pah1, and now they grow just fine on glycerol. And so when we took these endogenously truncated lipins and we put them into yeast, we could then recover this and restore this phenotype complemented. So this showed that they were indeed functional in cells. And then Valerie was able to actually purify tetrahymena thermophila Pah2. She biochemically characterized it to show that it was active and that an aspartate mutant in that active site reduced the activity. And then she was able to crystallize this domain, this protein, and determine its structure using selenomethionine SAD phasing. So just to back up, the key question was really, what is the function of the N-lip domain? And I briefly mentioned it, that the C-lip encodes for an enzyme domain called the haloacid dehalogenases. There's about 30,000 different members of this family, and they all phosphorylate different substrates, RNA, protein, and lipids. And what they share is structurally, they share this common scaffold shown here in blue. And the way that they generate specificity for their different substrates is with this unique cap region here in yellow that allows them to bind different types of molecules and dephosphorylate them. So our hypothesis going in was that the C-lip would encode for this HAD-like structure in blue, and this N-lip would actually be the cap region that generated specificity for phosphatidic acid. And when we saw the structure, we realized that we were completely wrong, but we found some very exciting things. So to walk you through it, we saw two domains. In pink here is the HAD-like domain of lipin, which encodes for the active site in the catalytic domain. And this was formed by the C-lip that we've color-coded in pink. And you can see that there was a calcium ion in the active site, which is occupying the position of magnesium, that these magnesium-dependent phosphatases use to dephosphorylate phosphatidic acid. And so we know that this was the site of the protein that would need to interact with the membrane. And then on left, we saw an immunoglobulin-like domain that was actually packing against this HAD-like domain to stabilize its structure. And what we thought was really, really interesting was that the N-lip, which we've color-coded here in blue, was actually not forming its own domain. It was actually co-folding with part of the C-lip to form this immunoglobulin domain. And so in Tt Pah2, the N and C-lip are only separated by about 2 residues, and those 2 residues are at the junction of this beta hairpin shown here. But remember, in humans and higher organisms, this 2 residues is going to be replaced by about 500 residues. And so the question is whether or not the N and C lip are always co-folded to form this immunoglobulin domain or not. And so we can sort of get a better picture of that if we use a surface view and separate out the N and C lip. Here we're showing them separated. Here on the left is the N lip. It sort of forms this horseshoe-like structure, and the C lip forms that catalytic domain, as well as this other sort of arms that wrap around the immunoglobulin, or the N lip, to form this immunoglobulin domain. So we have a hypothesis that we still need to test, is that potentially when this region, this linker, gets hyperphosphorylated, this could actually drive apart the N lip from the C lip to turn the enzyme off. And then when it gets dephosphorylated, it could reform this structure. But the other question that we wanted to answer was that how does lipin interact with the membrane? Remember, they're the only peripheral membrane enzyme in that glycerol-3 phosphate pathway. So how do they actually, what lipids do they recognize, what recruits them there, and how do they physically interact with them? And so the strategy we used was to collaborate with the Berk lab and use hydrogen deuterium exchange mass spectrometry. So if you're not familiar with this technique, it's a biophysical technique. And what you can do is you can take your protein and you can incubate it with heavy water that has deuterium. And the amide hydrogens in the peptide backbone will exchange with that deuterium. And that rate of exchange will be dependent on the solvent exposure of those hydrogens, so those residues, as well as the degree of secondary structure, such as alpha helices, which are stabilized by hydrogen bonds. So if you have alpha helices or beta strands, then the rate will be slower. And what we can do is we can identify conditions of the protein where they interact with artificial membranes called liposomes. And if they interact with the liposomes, when we ultracentrifuge them, that protein will pellet with the liposomes. And if the protein does not interact with those liposomes, then it will remain in the soluble phase. And so what you can do is you can take you can do this experiment, incubate it with deuterium, and then use mass spectrometry to map which regions exchange. You can do it with the protein alone, and then you can do it within the presence of liposomes that the protein binds. And you can therefore find regions where there's decreased exchange, and that could be either a region that interacts with the membrane or has formed a new secondary structure. So the first thing Valerie did was she found that Tt Pah2 interacts most strongly with liposomes with phosphatidic acid present. So here's just a neutral lipid with a PCE background. You see most of the protein is still soluble in the supernatant and very little is in the pellet with the liposome. But when we add in 20 mole% of phosphatidic acid, most of the protein is now present in the pellet, indicating a strong interaction. And then we did the HDX experiment with John Berk, and we found that there was 3 regions of the protein that were protected. Not surprisingly, one of the regions was the active site. We know that this region has to interact with the membrane to dephosphorylate phosphatidic acid. We also saw one peptide in that immunoglobulin domain that was protected, indicating that could also contribute to membrane binding. But the part that we actually saw that it was protected the most was actually a part that was disordered in our crystal structure. And the degree of protection that we saw was so drastic, we assumed that this is actually going from a disordered linker to forming an amphipathic helix in the presence of the membrane. So we think this is an N-terminal amphipathic helix. And this generated the hypothesis that this was the major region that was recruiting lipin to membranes. And so if we delete it, we should lose membrane binding. And so what Valerie did was she deleted that helix in Tt Pah2, and you can see that the wild type protein associates quite strongly with liposomes, but we have a complete loss of membrane binding when we delete that helix. So this amphipathic helix that's actually conserved between yeast and humans seems to be the major driver to membranes, or one of the major drivers. So you can read about that work. It was just published recently in Nature Communications. And now I want to tell you guys about a quick story about a new project coming out of the lab. And this is in the middle of that linker region, a region called the middle lipin domain. So this is the site of regulation for the protein. but we don't really know. It's not necessary for activity, but what about its regulation? And so what we noticed, I said most of it's intrinsically disordered, but we noticed through order disorder plots that there was about 100 residue region that was predicted to be ordered, and we called this the middle lipin domain. And this region is highly conserved among different homologs, and we hypothesized that it was playing some role in lipin's function. So what Xu did was she actually took that M-lip domain, the middle lipin domain, and deleted it. And we were able to purify both the full-length mouse lipin 1 as well as the full-length mouse lipin 1 where we've deleted that M-lip domain. She wanted to test if that's important for phosphatidic acid phosphatase activity, and she saw that surprisingly, the M-lip domain did not affect the enzyme activity of full-length mouse lipin 1. And this was using a tritoned mixed micelle-based type of assay. She was able to then test if it affected membrane binding using that liposome sedimentation assay. What she found was that indeed it does affect reduce membrane binding. Here we're looking at the full-length lipin, a very little bit of the protein pellets without a liposome. And when we add in just a neutral liposome with just PC, almost the majority of the protein starts to associate. And if we again add in 20 mole% of phosphatidic acid, almost all of the full-length protein is bound. In contrast, when we delete the M-lip domain, now in the neutral liposome with just PC, the majority of the protein is still soluble. And when we add phosphatidic acid, the majority of protein now still associates, but if we quantitate this, there's a significant and consistent reduction in membrane binding. So this suggested that the M-lip domain might be a new type of membrane binding domain. And what Wei Jing did was we wanted to understand how it might bind membranes. So he was able to actually purify just the isolated M-lip domain. And purifying this actually required detergents to extract it from E. coli membranes. And he tried to determine the structure of this domain, the full M-lip domain, but he wasn't able to. It wasn't very well behaved. But if he truncated off sort of this 15 residue C terminus that contains a number of highly conserved hydrophobic residues, tryptophans and phenylalanines, and basic residues, arginines and lysines, the protein now behaved extremely well, didn't require detergents, and he could actually determine the structure of the M-lip domain. And so he did this using selenomethionine SAD phasing, and here's the structure of what the M-lip domain is. And this was really exciting for us as a structural biologist because it revealed that the M-lip domain is actually a new protein fold that hadn't been previously reported. So this is a new way for a polypeptide chain to fold together into a three-dimensional structure. What we could tell from the structure was that the M-lip domain forms a very tightly associated dimer. So there's one subunit shown here in green and another subunit shown here in blue. And the dimer interface are they're forming this tightly associated dimer through these central alpha helices that form the majority of the contacts. Wei Jing actually was able to get structures of different crystal form of the mouse lipin domain. We also saw this tightly associated dimer, as well as crystal structures of the M-lip domain from mouse lipin 2. And it also formed this tightly associated dimer, almost an identical structure. And just to prove that it was a dimer in solution, we used a technique called size exclusion multi-angle light scattering that showed that the absolute molecular weight of this domain was consistent with the dimer in solution. And this raised an interesting possibility is that we know from other studies, previous studies, that mouse lipin 1 and 2 and 3 can actually homo and hetero dimerize with each other. And if we looked at that dimer interface, we actually noticed that the dimer interface was absolutely conserved between the different homologs. So all the residues at the dimer interface were identical between lipin 1, 2, and 3. And so this raised the possibility that this homo and hetero dimerization was occurring at least partially through this M-lip domain. And so we collaborated with Karen's lab, and she was actually able to show through co-IPs that deletion of the either the full M-lip domain or the M-lip domain that we were able to crystallize, this could reduce the association of lipin 1 with itself in homodimerization and actually more drastically affect the heterodimerization between lipin 1, 2, and 3. And lastly, I said we think it was actually affecting membrane binding. And so Wei Jing was actually able to show that in solution, that N-lip domain actually strongly associated with liposomes. If we have just a neutral one, about half of the protein is bound. But again, if we add in phosphatidic acid or other anionic lipids, now almost 100% of the protein is bound, and we've quantitated that down here. And we tested the effect of other lipids, and we didn't see any major effects. And if we delete that last C-terminal 15 residues, that the construct that we were able to crystallize, we have a little bit of membrane binding, but it's much, much less than when we have that C-terminal tail. And so we've also used HDXMS and to able to show that that C-terminal tail is the main driver, but other parts of that domain are actually interacting with the membrane as well. So with that, I'll just summarize some of the work that I showed you. Lipins contain these 3 domains, an N-lip, M, and C-lip. We know now that the N and C come together to form the sort of the business end of the molecule with the C-lip, the catalytic domain, and then they co-fold together to form this immunoglobulin domain. Lipins contain this N-terminal amphipathic helix, which is very critical for membrane binding. And we've been able to identify a new domain in lipins, this N-lip domain, which has its own set of residues that bind to membranes, but also at the same time simultaneously doubles the number of membrane binding sites through dimerization to increase its affinity for membranes. And with that, I just want to point out again everyone who did the work: Valerie, who worked on the Tt Pah2 structure, Wei Jing, and Xu Zhuan, who's not pictured here, and then our collaborators, Karen Rui and John Burke, as well as our funding sources. And I want to thank, again, the organizers and everyone for their attention, and I'd be happy to take any questions.